Okay, everybody, let's get to work here talking about some biology stuff. You guys realize, of course, that we're in the home stretch. Today's lecture and then just a couple more next week, and I'm out of your hair for a while, and at least until you find some other class that I'm teaching that you feel you must take. But this is good stuff, right? So far, I've been talking with you about prokaryotic and eukaryotic regulation of gene expression. That's been the topic for the last several lectures. I said at the end of lecture on Tuesday that I was going to save a little bit from that lecture, the little fine print about human genetics, and talk with you about that today, and then show you how the rubber hits the road in a very practical sense for a gene expression system, and talk with you about what's going on with these viruses. Okay, so before I get to talking about the viruses and how they game the gene expression system and get it to work in a way that the cells certainly wouldn't like, let me just say a few words to you about this human genetics business. So, remember from Tuesday, we're really complicated organisms. And we have all of these different cell types, but we've got just one set of instructions that needs to apply to all of them. And that gives rise to, that's a big problem, and it gives rise to a very complicated solution. There's all sorts of things that we do to play and control our gene expression. We package our DNA, we have very complicated RNA polymerases. We have lots of opportunities to modify the information content of our transcripts once they do get transcribed. They all get capped and polyadenylated, and almost effectively all of them get spliced. You can have alternative splicing, right? Just all of this stuff to deal with this big problem of tissue-specific regulation of gene expression. We also have a ridiculously large and complicated genome. So E. coli's genome, again, this very well understood, very well characterized prokaryotic organism that we've talked about so much now in this class, its genome is 4 million nucleotides long. Okay? And in the ballpark of 90 to 95% of those nucleotides are specifically spelling out instructions that get used by ribosomes to translate a protein, right? So pretty much all of E. coli's genome is what we would call an open reading frame, stretches of nucleotides that get translated by ribosomes after they've been transcribed. So keep that number in one side of your brain, right? 4 million nucleotide long genome, dense encoding information. And then on the other side, conjure up an image of our genome, 3.2 billion nucleotides long. Okay, so between friends, right? We've gotten to know each other pretty well in this class. You guys have gotten to know a lot more stuff about me than I have about you, and that's not fair. But our genome's roughly 1,000 times bigger than E. coli's genome, right? It's like 1,000 times bigger than E. coli's, and yet within our genome, the density of coding information is really, really small. It's on the order of 0.5% to 1%. So we still end up, when you put all those numbers together and you think about how much of coding information there is in E. coli versus coding information in a human, right? We still have about 10 times as much coding information as E. coli does. It's just that it's much less efficiently packaged and it sprawls out over vast distances. And our DNA, our genome, our chromosomes, for lack of a better word, it's a very good word, they're just filled with junk. And one of the ways that we know that a lot of the stuff in our genome is junk is because, first of all, it doesn't seem to cause people any problem if they don't have it. You can find people who have a chunk of DNA that's missing from one of their chromosomes, a few hundred base pairs here or there. No consequence, no manifestation, nothing different about them at all. It's also pretty easy to recognize as being junk because a lot of this junk is highly repeated. what's a good cartoon that I can talk with you guys about that you would relate to? For me, it's things like Calvin and Hobbes. You guys know what Calvin and Hobbes is? Okay, so let me use a Calvin and Hobbes sort of story. Calvin's the kind of, rebel kid. He gets an assignment in his school, write a book report. I want a 100-word report about War and Peace, right? And what does Calvin do? He goes home and he says, 100 words on War and Peace, this is going to be horrible. See, okay, the book was, I got 3 words, right? Very, 4 words. I got to come up with what? 96 more, 95 more. Okay, 1 more is going to be boring. And what he does is he fills in the remaining 95 by copying the word very over and over and over again, right? 95 times. The book was very, 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 very boring, right? There's his 100-word report. What kind of grade is Calvin going to get for that book report? It's going to be a pretty bad grade because there's just not that much information content there. He probably could have expressed, well, he certainly could have expressed the same sentiment by saying the book was very boring. If he wanted to really emphasize it, he could say it was very, very boring. If it was really important to get the idea across, he says it's very, very, very boring. But by the time you're at your 4th repeat, your 5th repeat, your 93rd repeat of that word, okay, I get the idea. This is just wasted space. This is junk. It's not very useful. So a lot of our genome we know is filled with junk because it's mutable and it doesn't cause any consequences when you don't have it as part of your chromosome for those people who have those types of deletions. And perhaps the biggest clue that it's junk is that it's highly repeated. There's a short stretch of nucleotide sequence within primate genomes, humans, chimps, gorillas, called an ALU repeat, ALU. It's about 250 nucleotides long. There are 1 million copies of that scattered randomly throughout the human genome. So if you guys thought that it was a stretch saying that a guy like Calvin might repeat the word very 95 times, well, come on, then nobody would do it. But how about our genome? We've got to stretch the equivalent 1 million copies of a 250 nucleotide long stretch of sequence. It's too short to code for a protein. It doesn't have any sort of promoter elements associated with it. It is junk, right? That accounts for about 10% of the human genome. That one repeat, 250 nucleotides long, 1 million copies, it's just shy of 10% of our 3.2 billion nucleotides. So our genomes are long, unwieldy, filled with lots of junk. Stop for a moment and think about the costs, the energetic costs. Think like a prokaryote for a moment here. Think about the energetic costs associated with all that junk. You derive no benefit from lugging around those 1 million copies of the ALU repeat, no appreciable benefit, and yet every time one of your cells divides, you've got to plow through and replicate that at a cost of 1 ATP per nucleotide. Right? So you're wasting millions of ATPs, 250 million, pretty close, every time you replicate your DNA just because you're lugging around that junk. So again, for prokaryotes, job number 1 is they keep their eye on the bottom line for energy. That's what's important to a prokaryote. And the rest sort of just comes as a result of focusing on the energetics. For eukaryotes, we're not nearly so worried about the energy. We're willing to throw away millions of ATPs to replicate junk when we replicate our chromosomes. We're willing to throw away millions of ATPs when we transcribe genes that have massive introns that never end up having anything at all to do with the coding sequence of the protein, the amino acids that get strung together to make a protein. And we're willing to throw away millions, tens of millions of ATPs making proteins sometimes that just never get delivered to the part of the cell or never get activated so that they're doing anything inside of the cell. All serving a different master. The energy isn't what's driving it. What's driving it for us is we want to make sure we express the right genes in the right tissues. That's an interesting difference in the philosophy between the two. And so that's an important thing to keep in mind when we're talking about the human genome and human genetics. But I also want to talk with you just a little bit now about the idea of using humans as a genetic organism. I've talked with you about this already in the context of Mendelian genetics. Remember the criteria that we talked about, or that I threw out at you, that made a good model genetic organism? Low upkeep, short generation time, large numbers of progeny, no ethical or moral constraints upon the type of experiments that you might want to do. Humans are like at the opposite end of the spectrum relative to things like pea plants and fruit flies and bacteria. For all of those criteria, and I say in the lecture notes, and I use my wife, her PhD is in human genetics. I used to kid her. It's been a while since this has come up. Maybe I need to talk with her about this tonight. But human geneticists are geneticists that deliberately, they go out of their way to study an organism where they have to do all of their work with one hand tied behind their back and a blindfold on. I mean, why would somebody choose to work in a system where you have these sorts of disadvantages? You know, you can't do forced mating experiments. You have to wait generations. You have to wait decades to see the result of your mating experiments. I mean, why would somebody choose to do that? Oh, yeah, there is an obvious answer. It's because it's about us, right? And it's because we can learn some things about us that might help improve the human condition. So that's why people do it. But boy, that's not what you would have thought would be the first choice for somebody who wanted to do genetics. An organism that has all these hang-ups for the mating experiments and such that need to be done for genetics. But it turns out that we can make about 250,000 different proteins over the course of our lives from the instructions we have within our chromosomes. And it's probably fair to say that for the vast majority of those 250,000 different proteins, if you mess one of them up, if you delete it, if you put in a premature stop codon, that sort of thing, if you mess one of them up, it will probably translate into some sort of genetic disease. All right? And we can talk about human genetic diseases where we know that's exactly what's happened. Cystic fibrosis. There's a single gene that if you get a lesion in that gene, if you mutate or delete or modify that gene, you get this disease that we call cystic fibrosis. Sickle cell anemia is another good example, right? Diabetes has a lot of genetic components to it. Cancer has a lot of genetic components to it. Almost all human genetic diseases, the ones that we understand well, at least at this point, can be traced to a defect in a single gene. And those genes get inherited in a Mendelian fashion, right? Just like the pea flower colors get inherited. Some of those mutations are dominant, some of those mutations are recessive, right? Depends on the different disease that you're talking about. And I think here's just an interesting idea for me to throw out to you guys. Maybe it'll be something that'll you'll be able to impress friends with at some point down the line. We all harbor a lot of messed up genes. Each of us does. On average, humans have 7 recessive lethal mutations. Okay? You know what those words mean. Put them all together, make sense of what I just said. Recessive lethal mutation. That means that if you had 2 bad copies, if you had 2 copies of that bad gene, dead, right? That's the lethal part. But you only have 1, and so you're not dead. If you had 2, you'd be dead. 7 on average, each of us has. There may be a couple of you, I'm not going to name names, that have 8 or 9. There may be a few that have only 5 or 6, right? But on average, 7 recessive lethals inside of each of us. There's a fancy genetic term to describe what I've just told you about. It's called genetic load. All right? The human genetic load is 7. The fruit fly genetic load is about 0.5. Right, so some populations have different genetic loads than others, but genetic load is ultimately why it is our society has a prohibition against incest. It's because my recessive lethals are pretty likely to be different from the recessive lethals of everybody else in this room. But they're pretty likely to be similar, 50% chance, to those of my sister's recessive lethals. it's just not a good idea to get those recessive lethals mixing together when there's such a good chance that you'll have that kind of overlap. And so incest is frowned upon largely because of that genetic load issue. And a question then seems to naturally arise is, well, what's up with that? Not the incest part, but why do we have why do we have a genetic load? Shouldn't this be a problem that takes care of itself, right? It doesn't take care of itself because these are recessives, so they don't manifest themselves unless you have two copies together, and they're rare, right? Mine are different than yours are different than mine. So it's hard to weed those out. It's hard to weed out recessive lethals because they don't, especially if they're uncommon, because they don't get together in a homozygous state that often. What about the dominant ones? What's up with that? I mentioned two just a little bit ago, cystic fibrosis and sickle cell anemia. Those are actually dominant mutations. Why does humanity still suffer from cystic fibrosis and sickle cell anemia? You know, cystic fibrosis is actually described in the Old Testament. It's been a pox, an affliction of humans for thousands of years, tens of thousands of years likely. What's up with that? Why can't this problem just take care of itself? Sorry, you got cystic fibrosis. You're not going to pass on your genes, and you know that means there will be less cystic fibrosis the next time around. Why do we still have it? The reason that we still have those types of dominant, lethal types of mutations or dominant mutations that give rise to disease, there's a word for that, or term for that. It's called heterozygote advantage. It turns out that individuals who have one bad copy of the cystic fibrosis gene and one good copy of the cystic fibrosis gene they develop symptoms of cystic fibrosis, but they tend to be mild, much less severe than if they were a homozygote. And while they have some symptoms of cystic fibrosis, they have resistance to certain respiratory types of infections that others don't have. And it's even clearer for sickle cell anemia because people with one bad copy of the sickle cell anemia gene have mild sickle cell anemia symptoms. It's kind of catchy. Yeah, that's a good idea. So if you have one bad copy of the sickle cell anemia gene, you have mild symptoms of sickle cell anemia, but you also get resistance to malaria. And so in certain parts of the world, resistance to malaria is a whole lot better cherry. It's a brass ring that you really like to have, and you're willing to pay the price, apparently, of having the mild sickle cell anemia symptoms, right? So why do we have these persistence? Why do we have this persistence of human genetic mutations, of the things that give rise to human genetic diseases? One of the reasons is because of just this genetic load idea. They're rare, and it's hard for the recessives to come together. And for the dominants, almost invariably, the dominant mutations have some sort of heterozygote advantage associated with them. And so you have to kind of look for the silver lining of having a mild case of cystic fibrosis. And where we live, a mild case of sickle cell anemia is where I wanted to go. Where we live, a mild case of sickle cell anemia, just not so good. You got sickle cell anemia. There's problems with that. In Africa, where malaria is common, you have a mild case of sickle cell anemia. Oh, okay, well, you know, you roll your dice, you take your chances. At least you're not going to get sleeping sickness, right? Look at the bright side. You're not going to get malaria, right? And so that's one of the reasons why we have this persistence of human genetic diseases, despite natural selection taken a heavy toll on human populations. And then lastly, about human genetics. One other thing that just might come in handy for you to know at some point down the line. I think this is an interesting fact. I talked with you a while back when I talked with you about karyotypes, about how if you can take a look at an individual's complete set of chromosomes, and that cytogeneticists who are trained to do this can do things like say, look at that, you've got a duplication, or you have an inversion of this part of one chromosome. And we've seen that before, and these are the sorts of things you should expect that this person is going to be afflicted with, or how they'll be different from other people who don't have that. In that context, I also told you that it was possible, easy actually, for these cytogeneticists to find aberrant chromosome numbers. That's a whole lot easier than looking at an inversion or a duplication of a part of a chromosome. And I specifically mentioned to you that trisomy of chromosome 21 was relatively common. And we all know what that is. That's Down syndrome, right? People with 3 copies of chromosome 21 have Down syndrome. What I didn't tell you, I mentioned it, but let me underscore it for you right now. Why is it that chromosome 21 is trisomic? It's not that there's something wrong with chromosome 21. What we are seeing is actually more of a manifestation of the fact that there's not that much genetic information on chromosome 21. It's a relatively small chromosome, right? They're numbered according to their size. Chromosome 1's the biggest, so it's relatively small. And having a messed up gene dosage for the genes on chromosome 21 doesn't have as much of a consequence as having a messed up gene dosage for pretty much every other chromosome. If you have 2 copies of chromosome 2, there's so many genes there and their expression levels are out of whack relative to all the rest of the genes within the genome that gives rise to a really serious problem that ultimately is incompatible with life. All right? There's some neat numbers that I put out for you in the lecture notes. You can look at the specifics if you like. There's like 4 or 5 chromosomes that are somewhat commonly seen in live births as being trisomic. Chromosome 18, 21, the sex chromosomes 22. Relatively small number. You never see the others because they're incompatible with life. 1 in 5 physicians will tell you 1 in 5 known pregnancies results in a spontaneous abortion because of a trisomy in the embryo, right? So it's a really common issue. And best guesses are that 1 in 2 of actual pregnancies are spontaneously aborted because of trisomy of 1 or more of the chromosomes, right? It's the ones that we see, the trisomy of chromosome 21, that's actually the relatively rare scenario. We've got all these other legions of fertilized eggs and embryos that we just never see because there's a spontaneous abortion before the woman even knows she's pregnant. So this trisomy thing, kind of a big deal, and that, it's hard to imagine how you would fix that. The basis of it is something that went wrong during the meiosis that gave rise to the gamete in either the male or the female. And boy, fixing that, I don't know. Personally, I can't imagine how you fix that. We may have that problem for quite a long time to come. All right, so there's a little spiel for you about human genetics. Strike home a little bit of what it is I've been talking about, as well as a few loose ends. Let me change gears a little bit and talk with you about viruses. So viruses have come up several times already during the course of this class. There's ongoing acrimonious debate between people as to are they alive or are they not, and are they cells that are they like your grandparents who have sold off their house and all of their cars and everything, they're simplifying their life so that they don't have as much complexity, or are they things that have independently just sort of figured out, boy, if I just have a simple approach I can game the system? Regardless of whether they're alive or not alive, however you want to fall in that debate, I want to talk with you today a little bit about how it is that viruses do what viruses need to do. The short answer is viruses do what they need to do by taking over a cell's gene expression apparatus. That's what it's all about. You know what a virus needs to do? A virus needs to make more copies of itself. Right? How does a virus make more copies of itself? It makes more copies of itself by taking over a cell's machinery, turning the cell's own infrastructure against itself so that the virus instead can take over the whole system. And so getting a glimpse at how it is that viruses play that game in the game that viruses are playing, I think maybe gives you a sense of how all-important the gene expression apparatus is. It's central, it's hugely important to what's going on for living things. So, again, we've already talked about viruses a number of times in this class. Here's a pretty picture that I think I've shown you before that shows you the variety of different kinds of viruses that afflict living things here on this planet. They have two things in common. I've already told you this, but let me remind you, all viruses have two things in common. They've got a nucleic acid genome that contains all the instructions that they need to be able to make more copies of themselves. And they have a protective outer protein coat. Right? Two things all viruses have in common. Now, I'm sure you remember and that you noticed that I said that they have a nucleic acid genome. That gives me some wiggle room because it's pretty much every kind of nucleic acid you might imagine. There are double-stranded DNA viruses. There are single-stranded DNA viruses. There are single-stranded RNA viruses, and there are even double-stranded RNA viruses. But all viruses have a nucleic acid genome. All of them have a protective outer protein coat. Some of them take things a step further. They have a phospholipid bilayer outside of that protective protein coat. And some of them are even a step further. In addition to the phospholipid bilayer, they'll have glycosylation and fancy sugary decorations on the outside of the phospholipid bilayer. Right? So there's a spectrum in terms of the complexity of viruses. But the name of the game for any virus, regardless if they're super simple, like these capsamere types, or these lunar lander modules, regardless, they all are playing the same game. That game, plain and simple, this is the game. All right? The game they're playing is that when the world comes to an end, they want to be the winner of the game, and the winner of the game is the thing that has the most copies of its DNA. That's the game that viruses are playing. And to some extent, you could argue that's the game that all living things are playing. But that is the victory criterion, right? At the end of the world, there's going to be a census, and whoever it is that has the most copies of their genes or the most copies of their chromosomes, that is declared the winner. I don't know what point there is in winning that game. The world's just come to an end. I don't know that I want to have a lot of copies of my genes. But nonetheless, that's the game that viruses are playing. It's all about optimizing or increasing the number of copies of their genetic material. Okay? And if that's the game that they're playing, that's the victory condition, that's the game they're playing, how do you work up a system that puts you in a good chance to win that game? That's the challenge that a virus has. Okay? It's all about that. So viruses are doing everything they can, and it's a red in tooth and claw kind of world for a virus. It's either you make a copy of yourself when you infect the cell, or you die. There's no gray area, like, oh, it didn't work this time, I'll try again tomorrow. When a virus interacts with certainly a prokaryotic cell, one of them is going to die and the other one's going to live. When a virus interacts with one of our cells, a bunch of our cells are going to die. Maybe a lot of our cells are going to die, but one way or the other, it's a life or death sort of struggle when they enter into this game, and the victor is the one that makes the most copies of themselves. So how is a virus going to pull that off? Let's talk about the first and most important step in playing this game. A virus has to be able to make a copy of itself, right? It needs to be able to find a suitable cell, infect that cell, take it over, and make copies of itself. So let's get an overview, just a big high-level picture, and I'll get into more detail about some of the specifics in the next couple of slides. Bottom line, a virus wants to make a bunch of copies of itself. It's first got to find a suitable cell to infect. then squirt its DNA in, or nucleic acid, if you want to be precise. You squirt the nucleic acid into the cell. That nucleic acid has to then get replicated, and you also have to use the information in that nucleic acid to make the protein coat and any other proteins that this particular virus might need. Then you need to assemble the virus into its, the nucleic acid wrapped up in its protein, and then you need to pull the trigger and bust open the cell's membrane to let the new baby viruses go out and do the process over again. Okay? That's it in a nutshell. That's what has to happen when a virus infects a bacterial cell. Take a moment, stop and think about how gene expression would come into play to accomplish, to be involved with that process. If you express the genes that the virus brings with it that breaks open the cell's wall, the membrane, at the very start, this is not going to work, right? If you bust open the cell before you've had a chance to make the other proteins that provide the protective coat for the virus, the nucleic acid, this is just stupid. You blew it, right? The virus can't express the genes that bust open the cell at the very onset, or the whole system's just not going to work. And by the same token, if you start expressing the genes that are involved with making the protein coat before you've made the genes or expressed the genes that take over the cell's gene expression apparatus, that's also probably not going to work. You're going to bundle up all of your nucleic acid before you've had a chance to express the genes off of it that take over the cell. Right? So here's the thing. The timing of the gene expression is really important for a virus. It can't just come in and have all of its genes being expressed at the same time. To be to be a player in this game, it behooves the virus to time things. And so you can't just squirt your DNA into the cell and have at it and start replicating and making proteins. There's got to be a sequence in which these events take place in order to optimize your chance to be able to win and take over. So again, there's some neat things that the virus has to do to be able to play this game. Not only does it have to take over the gene expression apparatus, get its genes expressed instead of the bacteria's genes, it needs to express its genes in a particular sequence in order to get the job done. So that's the overview. Let's take it a little more slowly, a little more detail, talking about how it is that this needs to play off. There's one of those cool lunar lander module guys, one of my favorite kinds of viruses. Let me just pause and take a moment to tell you a silly story. I was a postdoc before I came to Wright State at a place you guys have heard of. It's Harvard, right? So I hung out at Harvard for a little while. And there were guys that worked in the lab with me, right? Got to know them all quite well. Spent a lot of long hours in the lab when you're a postdoc. One of them, another postdoc, was this massive Swedish guy, Danny Lidholm, right? Massive Swedish guy. He was like 6'6" and probably 250 pounds, just really a big guy. Clearly, obviously, the descendant of Vikings. No question about it. He was proud that he had Viking blood, right? Big, booming voice. And another guy was this little, relatively small, I don't know, even that tiny. He was probably like 5'8", 5'7", something like that. Australian guy, real skinny, little Australian guy. His name's Alan Lowe. And Alan Lowe had this, we'd be working with viruses as part of our work, You could use them for cloning. I'm going to talk with you about that on Thursday. But Alan had this sort of annoying habit of using the British pronunciation of the alternative word for virus, right? So I've been careful so far to just use the word virus. You could also describe these viruses as phages, okay? The British pronunciation, and I suppose the Australian pronunciation, is phage, right? Danny Lidholm the Viking guy bought more into the American culture and the American pronunciation, and so he and Alan Lowe would go at it all the time. It's like, and Danny, this big booming Viking guy, would say, "It's phage, man. It's phage. It's not phage, it's phage, right? So for amongst us, I'm going to only use phage, but if you hear from somebody else, they're talking about phage, I'm worried that Danny Lidwam's going to storm into the room at some point, and if I slip and say phage, he's going to start screaming at me, and spit will come out, and all the rest of it. So bacteriophages, that thing up there, is more often referred to in the scientific literature as a phage or a bacteriophage than a virus. Check out what's going on for this bacteriophage, okay? It's got that lunar lander thing going, the legs coming down out there. You know what's up with the legs coming down? The legs coming down they're made out of protein, no surprise there. They're made out of protein, but those proteins have a job to do. It's not just aesthetically satisfying or makes it look pretty or cool. Those proteins are actually feeling around for things on the surface of cells that tell it, hey, this is the kind of cell that I can infect. You know how this works at a molecular level. There are things on the surface of the proteins that fit hand in glove or lock and key with things on the surface of the cells, right? There are proteins on the cells. There's proteins in the legs of the lunar lander modules. They stick to each other, and when they stick in just the right way, the virus gets the idea, hey, this is one of those cells that I should infect or that I could infect. What happens if a bacteriophage like that one, and I think that one's supposed to be bacteriophage T4, what if a bacteriophage T4 comes along and squirts its DNA into one of your cells? Not going to work. And why doesn't it work? I mean, this is a fatal error, literally, for the virus. If it squirts its DNA into one of your cells, why is it a fatal error? It's a fatal error because, for goodness sakes, you're a eukaryote. You have a whole different set of ways to turn genes on and off than prokaryotes do. Your promoter sequences are different, right? Remember? For prokaryotes, the promoters are at -35 and -10. For you, you've got something similar to the -10, but it's at -35. You got other stuff tens of thousands of base pairs away. What are the chances that a bacteriophage T4's genes will get expressed in one of your cells? Pretty much zero. It just doesn't work. The bacteriophage T4's promoters are promoters that have, through the pressures of evolution, have evolved to be spot on what a particular kind, a particular species of bacteria's gene expression apparatus is looking for. Spot on, right? And so it's not just any bacteria, it's got to be the bacteria for which its promoters are really set up to work. Bacteriophage T4, its host, the thing that it preys upon, is E. coli. And pretty much E. coli to the exclusion of everything else. It's the only thing that this virus ever goes after. How do you suppose bacteriophage T4 recognizes E. coli? It's got its little legs, it's feeling around. Just a bit of trivia, again, for the molecular biology Trivial Pursuit game that's coming out soon, right? Maybe you'll find it under your Christmas tree. What's it looking for? Just a bit of trivia. It's looking for P-lac Y, the lac permease protein. How about that? What an interesting coincidence, right? So it just happens that one of the proteins that we've been talking about is what it is the tips of the lunar lander module's legs are also looking for when it's contemplating. And P-lac Y is pretty much unique to E. coli. There's differences, there's similar things in other bacteria, but P-lac Y in E. coli has a particular amino acid sequence and structure such that T4 can recognize it and bind to it.
So does that mean it would be easier for T4 to find E. coli and infect them in a lactose environment where E. coli are making more?
Absolutely, absolutely. So T4 has got a better shot in it. If you want to get E. coli infected by T4, and people do this in the lab, right? There are experiments that you might do. You want to also have lactose in the environment, and you may want to even starve it for glucose so you have a good chance of getting the lac operon expressed. How about that? little bit of trivia for you. So, first step in the process is you have to have an infection. This is what infection's all about. You get the virus, it sticks to a specific set of receptors that it's looking for on the surface of the bacteria. And then, you know, in the past, I've said that's when the virus shoots its wad. And that always generates a lot of laughter. It makes me think that there's something going on when I say that I don't intend. So I'm going to just forget I said that. It squirts its DNA into the cell. Once it's checked it out, it feels, oh, this is the kind of cell I want to infect, then, and only then, does the nucleic acid go in, right? Because if you squirt your DNA into a cell that you're not set up to take over, fatal error, the virus loses, the bacteria says, what was that? And it just goes about its business, right? So somebody gave me a free lunch of some nucleic acid. I didn't even have to go out and eat it. Just voila, here it is. There's DNA that's spontaneously being thrown at me for no apparent reason. But let's say this is the kind of cell that phage should be infecting. What do you want to have happen next? If you're the virus, what's your plan? You want to take over that cell's gene expression apparatus. Think of it as like a military operation, right? If you want to take out an opposing, an enemy, what do you go for first? You know, it makes good sense. It's a good strategy to take out the brains of the operation, the command center, right? Ruin the ability, to compromise the ability of the thing you're attacking to communicate with the troops out in the field. A lot of viruses, the first thing that'll happen is among the first genes to be expressed are those that specifically chew up the host cell's DNA. Because if you take out the DNA, you know, that gives you more time then to do the mop-up operation and take over the rest of the gene expression apparatus. And what do you suppose the bacteria's first line of defense is? It's the same thing. It's got proteins present within its cytoplasm whose job it is to chew up DNA that's not its own. And it can do that because there's modifications that can be made to the DNA methylation. And so each bacteria will methylate its DNA a little bit differently. Its proteins say, oh, that's my methylation pattern. I don't chew up anything that has this methylation pattern. But a virus, when it comes in, often won't have the right methylation pattern on it, and it can get chewed up. So there's a battle. First thing to go is you try to take the DNA out of the picture. And then if you get away with that as a virus, then the next thing to do is, okay, now let's sit back. We got some time. Let's make the proteins that we're going to need to protect the virus once we make new baby copies of the virus, and let's do the other things that need to be done. If you need to replicate the viral DNA, those sorts of things will be expressed and be allowed to happen. But the way that ultimately happens is the virus has to be more appealing to the RNA polymerases and the ribosomes of the virus, of the cell. It needs to be more appealing. Okay, let me just start fresh. The virus has to have promoters and translational start sites that are more appealing to the cell's RNA polymerases and ribosomes than the cell's own promoters and translational start sites are. Right? So again, there's this pressure, evolutionary pressure on the viruses to really get good matches to what sort of nucleotides those sorts of things are looking for, the RNA polymerases and the ribosomes. And then once you've made all of those proteins, then you can start to assemble the viruses. And for these bacteriophages, that's actually a pretty convoluted process in and of itself. You can see there's 3 simultaneous things that are going on for the assembly. It's kind of like putting together a plastic model of a boat or something for a hobby. And it's only at that point that it becomes reasonable and appropriate for the virus to bust open the cell and try and take its chances out in the world again and see if it can repeat this process. This picture, this cartoon, isn't that far off the mark in an interesting aspect here. Look at how many viruses bust out of that cell. So 5, right? When a bacteriophage T4 infects an E. coli cell, the average yield, the payoff for a successful infection, is 5 to 10 new viruses. So for every bacteria that dies, you make only 5 to 10 new viruses. For another E. coli bacteria, bacteriophage lambda, the tally's a little bit higher, but it's only about 40 viruses get produced. So it's not like when a virus infects a cell, you get this Vegas-style slot machine payoff of, they just keep spewing out, You get a handful of new viruses that can go out and do the whole thing again. Right? What's the name of the game? The name of the game is the virus wins that has made the most copies of itself and for which there are the most copies when the world comes to an end. Okay? This whole process in a bacteria takes anywhere from 30 to 60 minutes. Right? Every 30 to 60 minutes, a virus can infect a cell, take it over, and then make 5 to 50 copies of itself, depending on the virus that you're talking about. Right? That's the game. That's what they're trying to do. Nice strategy, right? Could you do better? Because if you want to be a winning virus, maybe you should stop and think, okay, I got it. I got this system down. This works. Is there a way to do it better? I'll tell you, there is. What I've just described to you is what's called a lytic phage. Lytic, it's lysing. Lytic, lysing. It's busting open the cell every time it infects it. Right? There's no subtlety. There's no clever strategy beyond what it is I've talked with you about here. It's kind of brute force. It's brutish. It just comes in, busts things up, and goes out and tries to do it again. Not a very clever or sophisticated strategy beyond what it is that it's actually accomplishing. And you get a payoff. You get maybe 5, 10, 50 new viruses for adopting this strategy of kill every time you infect the cell. But let me tell you about an alternative that's more subtle, more diabolical. A lysogenic phage. So here, again, is the lytic phage. You infect, you take over, you make copies of yourself, you bust things up, and out you go. Payoff of 5, let's say 50, wildly optimistic, you got 50 copies. Here is an alternative called a lysogenic cycle. The essence of a lysogenic phage, how a lysogenic phage differs from a lytic phage, is this. A lysogenic phage is checking out how the cell is doing. If the cell that it has infected looks like it's fat and happy, the lysogenic phage says, this is a fat and happy cell. Why would I want to go out into the cold, hard world where I might go weeks, months, decades, maybe even centuries, I'm not joking, maybe centuries before I'm going to find another cell that I can infect? Right? This cell's fat and happy. It's doing great. It's comfortable in here. Who knows when the next time I'm going to find another cell is? Right? This one's doing great. How about if I just hitch my cart to this wagon? That's wrong, right? How about if I hitch my cart to this horse and let this horse, I'm having a lot of trouble with this class with cows and horses and barnyard sorts of things, but why don't I just hitch a ride, right? I'm going to be clever about this. Every time this fat and happy cell replicates, I am going to replicate along with it. And I don't have to fend for myself out in the world. I don't have to worry about finding another cell to infect. I got one. Every time it divides, and that's going to be what? Fat and happy, right? 30 minutes or so, there'll be another pair of these cells. And instead of having a virus that infects a cell and gets you 50 an hour later, 50 new viruses an hour later, now what we've got is a virus that infects the cell, and a half hour later, where once there was one cell infected by this virus. Now there are 2 cells infected with this virus. It's a long-term strategy because, again, it may be a long time before those 50 find another cell that they can infect, but another half hour later, those 2 infected cells become 4 infected cells. So at an hour, we've got a payoff of 4 as opposed to 50. An hour and a half, we got 8. 2 hours, we got 16. All right, 2.5 hours, we're all the way up to 32. 3 hours, we're up to what? 64. We've already done better than if we had simply infected that cell, pulled the trigger, gotten our 50 out, unless those 50 were also able to go out and infect some other cells. All right? So it's a more guaranteed approach if you're infecting a fat, happy cell where it's got no problems, just hang out in that cell. Don't kill it. But, here's the important but, when the cell that you've infected isn't so fat and happy anymore, come on, it's going to happen eventually. When it's not that fat and happy anymore, then you kill it. Then you switch gears and you go from the lysogenic sort of strategy to a lytic strategy, and you get the payoff you would have had if you had pulled the trigger right out of the gate. But you get it on a much larger population of cells than if you had done it right out of the gate. How about that for diabolical? And so for a lysogenic phage, what it's doing is that immediately upon infection, it checks out how the cell is doing. Is this cell fair and all right or not? If it gets the idea that, well, it's low on ATP, it's suffered from exposure to heat, it's starving for nitrogen, if any of a number of things are detected, the virus says, okay, look, we're going to pull the trigger, put this thing out of its misery, I'm going to take over, make copies of myself, and like a rat, I'm out of this sinking ship and off to see if I can find a new ship that I can take over down the line. I'll take my chances out in the cold, hard world. Right, so it picks up, it's sensing, it looks for the conditions inside of the cell. But if it gets the idea that, look at this, there's an awful lot of ATP here, there's a lot of glucose, there's no indications of damage from heat, nitrogen levels seem fine, all this stuff looks pretty good, what this lysogenic virus now will do is it will integrate its DNA, its chromosome, into the cell's chromosome and kind of hitch a ride. Every time the cell's chromosome replicates, the virus replicates along with it. So each round of cell division, you end up with twice the number of chromosomes that have that virus integrated into them. And the process just goes over and over again. If there's a signal at some point that says, hey, wait, glucose levels are low, what do? You pop the virus out, you enter into the lysogenic phage, and you go down that path over there. How about that for diabolical, for clever, right? Again, the name of the game is make as many copies of yourself as is possible. So many viruses are fairly unsophisticated. They can only be lytic. They're lytic phages, that's it. But some of them have taken things to another level, and they're not. They can vacillate. They can switch back and forth between being lysogenic and being lytic. Right? That's an even more diabolical strategy. And again, ultimately, it's all accomplished through manipulating the gene expression system. Okay, so we're through this part of the outline. Let me say a little bit to you about the evolution of viruses. Viruses have clearly been around for a very long time, like billions of years. And I find this fascinating. You all know about the measles virus, right? Right? I'm probably the only person you'll ever encounter. One more strange thing you get to learn about me in this class. I'm the only guy you'll probably ever meet that got infected by measles twice. Right? So how about that? So I've had a double shot of measles. I'm almost, well, and chicken pox I got a couple times too. So nonetheless, the measles virus, right? plagued humanity for a long time. You guys are probably all immune. You got a vaccine. There wasn't one when I was in the age where I could have benefited from it. But the measles virus that is a pox upon humans for millennia turns out to be evolutionarily related to some viruses that also infect cows and sheep and deer. And it's evolutionarily related to a virus that infects cetaceans, the dolphins, and it's also evolutionarily related to a virus, the distemper virus. So now you know measles is the human equivalent of a dog's distemper. How about that? So these viruses probably started by infecting a single common ancestor to all of these different mammals. And then as the different mammals evolved and diverged relative to each other, the viruses, the little cloud of viral particles that traveled along with them, also evolved, so that now they're best adapted to infecting humans. And the human measles virus has very little prospect of doing anything of consequence when it infects a dog. Why? Well, because there's been divergent evolution, and the measles virus is geared up to be able to infect and take over our cells. It's just not quite so good a match for what you need to do to accomplish the same thing in a dog or a bear or a cat. All right? So that's, again, it's an interesting evolutionary history. And then, related to that evolutionary history, I know you've all heard about AIDS, the acquired immune deficiency syndrome caused by a single virus called human immunodeficiency virus, HIV. All right? Neat things have been done to figure out, where did this thing come from? What's up with that? Because HIV, for all intents and purposes, seems to be relatively new under the sun. You know, in the 50s, there was no such thing as AIDS, right? Nothing. No, and then all of a sudden, going back to like the 70s, suddenly people started to get this syndrome, and there was an intensive effort to figure out, what's this? What's this new disease? Well, it looks very much as if what happened is that some people in Africa ate some monkeys that were infected with a virus that we now call SIV, simian immunodeficiency virus. And the monkeys, the virus that was infecting the monkey was close enough to what it needed to get done to be able to infect a human. And then we got this new infection, and then other humans got infected by the human who had it. And you can see this sort of evolutionary relationship then. And it looks as if there may have been two independent origins of HIV. One that gave rise to this group, the HIV-1M group, and the one that gave to HIV-2. There may have been two times that a human got infected by the virus. Interesting stray thought about viruses. HIV isn't a very good virus in an important sense. Remember the name of the game? The name of the game is make as many copies of yourself so that when the world comes to an end and they count noses, or capsids, I suppose, is the relevant term for viruses. When they do that census, you want to be the one that has the biggest number. Killing the thing that you infect, not a really good strategy. You saw that for the lysogenic phage, right? It's much better if you just infect, you lay low, and you ride it out. You make copies, right? You know what the best strategy for a virus is? For a virus that infects humans in particular? The very best are the ones at playing this game are the ones that infect you, and you don't even know that you've been infected, right? Doesn't slow you down a bit. You just go back out, you get infected, all right, I got it. Now I'm going to go out into the world and I infect others that I bump into and that I encounter. And they don't know that they got infected. And they infect others. And eventually everybody gets infected, right? I got one of those viruses, turns out. 95% of people in North America have one of those viruses. You get infected, you don't even know it. But if you donate blood, one of the things they check to see is if you've got this virus in you, and if you do, they don't give you your blood to somebody who hasn't been infected with the virus because we just don't know. But that's a standard test for anybody who donates blood. They'll come back and they'll tell you if you look. Off the top of my head, I don't recall, but I'll find out for you. So, how about that? That's the strategy that you want to have. And you know what? HIV, over the course of just, 30, 40 years of infecting people, shows serious movement toward being that kind of virus. It's evolving, it's adapting, so that people who get infected with HIV don't die right away. They hang around for a while so they can infect other people. How about that? This virus is playing the game just like a lysogenic phage would. And part of the thing that's such a disaster about HIV is that we got infected by an amateur. All right? If only it had been a more sophisticated virus like the one that gives you a cold. Okay, you're out of action for a few days. Or the flu. Okay, you're out of action for a week. But you can still, you survive, you spread your virus around. HIV, not quite a good match, because you know what? In monkeys don't display much disease at all when they're infected with SIV, because SIV is much better adapted for life inside of monkeys than HIV is adapted for life inside of us. All right. So, if you're curious, this is what's going on with a retrovirus like HIV. Here, this is a pretty sophisticated one. It's got the glycoproteins on the outside. It's got the lipids. It fuses. First thing that has to happen is you have to make a DNA copy of the RNA genome. And then the rest is pretty much the same as what we had talked about before. This table is straight out of your textbook. The one on the next slide is also straight out of your textbook. That's just in case you're curious about all the different categories and kinds of viruses. I'm not going to ask you specific questions about that on the exam. And here's a really pretty picture, right? Real world life and death struggle. You can see some of those bacteriophages trying to take over that cell. Well, let's see what happens if I click on that.
Really unexpected surprise right before your eyes. On the court, we don't compromise with our sick man that stands cheering us on. We build plans. We create memories together. We strive for victories together. We go hard, hustling for the dream, standing tall with pride. We rate her up. We're all one.
That's cool.
We're your team.
How about that? So did you guys know that the basketball game at Wright State on Friday, tomorrow night? And they are really hoping we'll have a lot of Wright State students there.
A Sunday matinee feature
I don't want this there.
boys basketball programs in the state. Second ranked New North travel north to Danvers to take on number.
There. How about that? So, how about that? You know, a big part of going to a college, major Division I universities, you get to go to some neat things, basketball games and such. So, there's somebody up there in the back of the room that I think wants to say something about that. How about that? You guys recognize Rowdy? That's Wright State's mascot, and he's here as a surprise visit today to help encourage you guys to come to the game on Friday night. How about that? Here's Rowdy, and Rowdy, you got some stuff to give to these folks, don't you? I'm going to get out of the way and let Rowdy take over.
We're still doing instruction, and I was like, oh no!
So they're all yours.